Hi, this is E. David Crawford, Editor-in-Chief of Grand Rounds in Urology. Over the years, we have relied on standard sort of agar plate culture and sensitivities to determine the presence or absence of infection. Most recently, there's been a new way to look at that involves next-generation sequencing. Joining me is one of the international experts in infections in urology. He's also well known for his work with implantable penile prosthesis, probably does more than 200 a year. He did his training at Duke and then spent 3 years with Dr. Steve Wilson. And so Henry is going to spend a few minutes here telling us a little bit about next generation sequencing, why and where to use it, and the difference between that and our standard culture, and then some of his work with biofilms and penile implants. So, Gerard, thanks for joining us.
Thank you, Dr. Crawford. We'll go ahead and get right into it. Next generation sequencing of DNA is what we're talking about. This is going to investigate the bacteria and fungus that is at your patient's in vivo sites for both urine and penile implants. And we'll also look at prostatic secretions. It is the gold standard for identifying isolates for wound healing and starting to be for orthopedic joint infections. So this is what led me to start investigating it. I've already written a lot of papers on biofilm and bacterial infections and penile implants. I was reading other journal articles and I found this out. This data has already won 2 best clinical abstracts at national level meetings in both 2018 and 19. And we have an AUA abstract this year in 2020 that we'll talk about later in the presentation. Basically, next generation sequencing gives significantly more data than traditional cultures. All right, so as I alluded to in the last slide, in the field of urology, it can be applied to 3 main areas. There's probably more, but one of them, I think the biggest, is urinary tract infections. If you're having troubles with diagnosing your patient, next-gen sequencing appears to be much better than traditional cultures for urinary tract infections. Prostatic infections, Curtis Nickel is looking at this and how it interacts with better diagnosis for prostatic infections. And of course, I'm going to present mainly on penile implant infections. It's reimbursed by Medicare and many commercial insurance plans. Why next-gen sequencing? All right, so the physician orders the test. If you have the PCR available at your facility, you get the initial PCR data back immediately, like within an hour or 2. So you get immediate results, not having to wait a long time. This is the PCR data, it's the quick data. Then you send it to Lubbock, Texas for where it goes through next-gen sequencing, and that can take 3 to 5 days once it arrives there for you to get the report back. We are going to go over those reports and how it can help benefit you in your lab. All right, so here is a report. This is the next-gen sequencing. It's very busy, so we're going to take a minute here. One very important data is that you get, step 1 is you look at what antibiotics could be resistant in your patient's data right there, step 1. Step 2 is what bacteria, here E. coli. One big advantage of next-gen sequencing over traditional culturing is what I call abundance. Not only does it list more bacteria, what bacteria is there, but also the abundance. Abundance is very important for clinical health. So you may be culturing staph for a penile implant, but that'll be a very low load, as we're going to see in a later example. But another bacteria is actually what's causing the infection. So look at bacterial load, or what I call abundance. You can also have fungus. 15% of penile implant infections now we think are fungus related. not bacteria related. And then step 4, of course, is looking at what antibiotics can be used or antifungals for your patient's infection. So go through it very slowly. It takes a few reports to become comfortable, but look at abundance and, of course, resistance. And then here it tells you what you can prescribe the patient to help you. All right, so we're going to go straight to a urinary tract infection patient. The patient has been using traditional cultures and been finding E. coli at low colony counts. The patient was treated for E. coli. The patient did not respond well. The patient then underwent next-gen sequencing. It showed, yes, E. coli was there, but at a very low abundance. Another bacteria is what's causing the infection. This is not something that's known to urologists. I'm not even sure exactly how to pronounce it, but I call it leptotrichia. So thereby, it's not E. coli causing this patient's symptoms, it's leptotrichia. And then over here, they'll give you the antibiotic that's proper to treat that, and the patient recovered. So this is an excellent example of what traditional cultures have been showing, and then how next-gen sequencing is showing you the correct or the more abundant bacteria that's there. All right, here's one of my patients. It's a 72-year-old patient, 3 months after a penile implant. He was diabetic. He presented with an open incision with draining gross pus. One thing that's interesting is 50% of patients are culture negative with traditional cultures. That's been repeatedly shown in the literature. This patient did not have any bacteria show up on his traditional culture. However, he had gross pus draining from his incision. While the traditional culture came back negative, the next-gen sequencing came back with Morganella with 99%. I think that next-gen sequencing is going to be rewriting what pathogens are actually causing bacterial infections. Apparently, staph grows very easy on agar plates, and other bacteria like Morganella does not. That is my knowledge of agar plates. But I have another patient who had Actinobacter. It was culture negative, but next-gen sequencing caused this. It's a bacteria I didn't know existed, and now it's going to be in a huge abundance on several patients that we're going to present. So here is the 2020 AUA abstract that was accepted for podium presentation. I want you to note, is this the new gold standard? I think it's becoming the new gold standard. So in this, sequencing of DNA versus traditional cultures in vivo. So as I stated earlier, previous studies have used traditional culture methods to identify the microbial species presence at the time of removal and replacement of penile implant.
So, well, thanks, Dr. Henry. That was very interesting and really is the future. So explain this a little bit in more depth. PCR is utilized and can be done if you have the reagents within your office to identify some of the more common bacteria. Is that correct?
Yes, sir. It would definitely give you a quick, rapid response. That's the idea behind it. I would almost compare it to a gram stain in terms of how quick you can get the data back, and it can show you the major pathogens within 2 hours.
So it's like the more common pathogens, 25 to 30 or something like that, correct? Is that
yes, sir.
The way it works? But what happens is we know there's more pathogens, and the information that's provided by when you send it off next generation sequencing, you can identify, I've heard, up 25 or 30,000 different types of bacteria and fungal species. Is that your understanding also?
Yeah, I've heard more than 40,000. Yeah, it's
wow. it grows every day. So
Yes.
that's interesting, and I think as you alluded to, we're finding that many of these infections that we don't understand them. We're finding that there may be bacteria or fungal origin, dating things like interstitial cystitis and prostatitis and infections that are relapsing. So, but you had spent probably more time than anybody with penile prosthesis and infections and biofilm. So, how would we define a biofilm, and how have you found that next-gen sequencing and this helps you there just a little bit?
Sure, of course. Well, I do think, as I alluded earlier, I think the biggest use for this in urology, in urinary tract infections. For penile implants, I think one of the things it's going to do is rewrite what are actually causing the pathogen that are actually causing the infections. Biofilm was first really discussed by Dr. Costerton, who is kind of almost in some ways the beginning of not only biofilm, but in some ways this actual company that's doing next-gen sequencing, Microgen. And he described it basically as a area where bacteria accumulate, they first attach, and then they shield themselves from the host defenses, like, white cells from antibiotics and other host defense mechanisms. So biofilm is the bacteria attach, they colonate, and they form a shield matrix where they hide behind. So most antibiotics attack planktonic or swimming around bacteria. They can't really attack bacteria in the biofilm because they're behind that shield. And so biofilm is basically on all implants. It doesn't matter if it's a penile implant, breast implant, hip, knee, heart valve. Biofilm occurs on anything you put inside the body and hides there. When you reoperate, it can cause increased infection rates. It's basically growing at a very low growth rate. And that's from Dr. Costerton, who kind of started this lab in Lubbock, Texas.
Uh-huh. So, so in the biofilm and infected prosthesis, the next-gen sequencing helps you identify what the organisms are that would not have been so easy to identify with standard culture. Is that
or not being identified by standard culture? Not only helping it, it's when we get to our data here, it's basically showing statistically significant not only numbers of bacteria, but bacteria I haven't even seen before on cultures. And I have a Excel data sheet of over 400 patients with bacterial cultures, and I'm finding brand new bacteria, several bacteria. But one of the things I love, as I alluded to earlier, is that the abundance tells me exactly which one appears to be actually causing the infection.
That's really exciting. I think that this next-gen sequencing is going to continue to be a real game changer in urologic infections and understanding etiologies of a number of enigmas that we deal with diseases there. So thanks for your time, and we look forward to hearing your presentation at the AUA, which will be virtual this year.